everyone, I'm Annika and welcome back to the Chat Lab. Today I am so excited to introduce Professor Thomas Silhavy from Princeton University. He is a leading molecular biologist for groundbreaking work on how bacteria build and maintain their outer membranes, discoveries that have helped shape modern microbiology and helped guide new directions in antibiotic research. I loved our conversation. He was incredibly knowledgeable, yet so kind and easy to talk to. I'm really grateful for the time he took to speak with me and share his expertise with me. Please welcome Professor Silhavy. Dr. Silhavy, I'm so very honored to have you on the Chat Lab. Your work has changed the way we think about bacterial cell biology, and I cannot wait to get into the science, the stories, and everything that has shaped your journey. Thank you so much for joining me again today.
My pleasure.
So, to start us off, I'd love to hear about the beginning of your journey. I mean, what first pulled you into this world of molecular biology and bacteria?
I actually went to pharmacy school at a place called Ferris State University. I really I really think that pharmacists are vastly overeducated for what they do. So I found that profession kind of frustrating.
Mhm.
So like most of my colleagues, I got started in research as an undergraduate. Most of what I did was chemistry, but my advisor convinced me to go into biochemistry because he thought the future of biochemistry was better than the future of chemistry. All right.
Interesting.
So Ferris is in Michigan, and so I applied to a bunch of Big Ten schools for graduate school. But my advisor convinced me to apply to a couple of the big ones,
Mm.
like Harvard and Stanford. And I bet him a bottle of scotch that I would never get into those places, but I did. So Harvard took a chance on me, and I went to Harvard for
Wow.
graduate school. And I did start in biochemistry. But when I was in graduate school, I took a course in bacterial genetics, and I fell in love with it. And so I did my postdoc in bacterial genetics. My thesis advisor was a German, and he was scheduled to take a sabbatical at the Institut Pasteur in Paris. But before that started, he got offered a job in Germany at the University of Constance. So he had to take me with him when he left. So I spent the last year or so of my graduate career in Europe, first at the Institut Pasteur and then at the University of Constance. Yeah, that was a wonderful experience, actually.
Yeah, I can imagine. I mean, when you look back at that early experience, that moment that really pulled you in, I mean, was there a particular mentor or experiment or challenge that really kind of solidified your decision to stay in that field of biochem?
Well, like I told you, um, I I found pharmacy kind of boring.
Mm,
So if I had a chance to go to graduate school, I thought, what the heck? I'll go to graduate school.
wow.
If it doesn't
Okay.
work, I can always be a pharmacist.
That's true. I mean, I love hearing about those early experiences that shaped your path. I mean, it really shows how mentorship and curiosity can change a career. Building on that, once you found your footing in molecular biology, what was the first big challenge or puzzle in the lab?
As a graduate student, I worked on sugar transport systems in bacteria.
Wow.
In particular, the galactose transport system and the maltose transport system.
Mm.
So you know E. coli has two membranes. In the maltose transport system, there's 5 proteins, one in the outer membrane, one in the periplasm between the 2 membranes, and 2 in the membrane and 1 in the cytoplasm. So it's what's called an ATP, an ABC transporter. It's
Oh,
an ATP binding cassette transporter.
interesting.
Okay, it's one of the best characterized now, actually.
Wow.
But at the time we were working on this, it wasn't clear how a soluble periplasmic protein could be involved in transport. So that was a big focus of what we did, was to actually prove that they really are required and get some insight into what it is that they do.
In those early years, molecular biology was evolving so quickly. I mean, was there a particular technique or technological shift, like of recombinant DNA or new genetic tools that changed the way you approached your research?
Well, my postdoc was involved using gene fusions. I don't, so we would take parts of proteins and fuse them to beta galactosidase.
Mm.
But this was before there was recombinant DNA.
Oh, okay.
So we did it using transposable genetic elements.
Interesting. So, hearing about those early turning points really sets the stage for the incredible work that you went on to do. I mean, your lab has made some of the first discoveries about signal sequences and how proteins were to go inside a bacterial cell. So, for people who aren't specialists or for high schoolers like me, how would you describe why that discovery even mattered and what it revealed about how bacteria are organized?
Well, when I was a graduate student, we knew that there was an inner membrane, an outer membrane, and a periplasm. We knew that.
Mm.
And as I told you, I was working on periplasmic proteins. So as a postdoc, I went to John Beckwith's lab because he was interested in the question, how do you get a protein out of the cytoplasm? Okay. And he suggested using gene fusions.
Mm.
So if you fuse something like maltose binding protein at the amino terminus and beta galactosidase at the C terminus, the cell tries to secrete that protein.
Oh.
But the beta galactosidase folds faster than it can be secreted, and it gets stuck in the secretion machinery. And it
Mm.
blocks all protein secretion. And the poor constipated bacteria dies. Okay.
Okay.
Now, if you put a signal sequence mutation in the hybrid protein, then the cell will not try and export it and it won't die. So that's how we did it.
Wow, that's amazing.
The hybrid protein was only made when it was induced. So
And
since it was maltose binding protein, it was induced with maltose.
maltose.
It was perfectly happy unless you added maltose.
Oh, so that was kind of inhibiting the process entirely.
Yeah.
Got it. Okay, so once you've uncovered how these signal sequences guide proteins, how did that discovery shape the next big question your lab wanted to tackle?
The the next big question is who's recognizing the signal sequence? And so we looked for mutations that restored recognition.
Mm,
So these are called suppressor mutations.
interesting.
So we altered the machine that recognizes the signal sequence by suppressor mutation. That's how we identified the machine.
So, and as you were exploring this protein targeting, did you ever encounter results that completely contradicted what the field assumed at the time?
No, what actually happened is it sort of reinforced what was happening at the time.
Oh, wow.
Because at the time we were working with bacteria, Gunther Blobel was working with mammalian cells.
Mm,
And he won the Nobel Prize for the signal hypothesis.
okay.
But our data with bacteria provided the first direct evidence that signal sequences were required, and it indicated that signal sequences work in all organisms.
So, stepping back from the early discoveries, what do you think people often misunderstand about the outer membrane and why it is such a critical structure for bacterial survival?
I'm not sure that most people know that there is one. I mean, there's two major classes of bacteria, right? Gram
Right.
positives that don't have an outer membrane, and
And
gram
gram-negative
negatives that
then
do.
too.
And what is certainly true is that gram-negative bacteria are almost always more resistant to antibiotics than gram-positive bacteria.
Mm,
because of the outer membrane is a very effective permeability barrier.
right.
So some people understand that.
Building on that, one thing your lab is known for is uncovering the machinery that assembles this outer membrane. I mean, what I'm curious about is how you first recognized that this machinery even existed.
It has to exist.
Was there a specific clue or experimental result that made you realize that there was a dedicated system behind it?
Yes. So, um, I mean, the thing that's always fascinated me about the outer membrane is that it's built on the other side of the cell wall in an environment that doesn't have any energy,
Mm,
right?
right.
And so there was a mutation that was called IMP for increased permeability. Okay. And so this strain still made an outer membrane, but the permeability barrier was totally destroyed.
Mm.
So it was very sensitive to all kinds of stuff.
This is so interesting. I'm learning so much already.
Well,
Given everything we know about the outer membrane assembly, how do you see this knowledge shaping this research for new antibiotics, especially as this resistance continues to rise now?
well, the components in the outer membrane that build the outer membrane are essential. All right. You
Hmm.
can't take them away.
Right.
So there are antibiotics now that target those proteins.
Oh.
All right. And the nice thing about that is that since those proteins are on the surface, it doesn't matter. The outer membrane permeability barrier is irrelevant.
Oh, that makes sense.
Okay. So you can actually kill a gram-negative bacteria without the compound crossing any membrane at all.
Oh, wow.
So those exist now, but
Mm.
getting that getting those to the into drug form, that's a long, complicated process.
Yeah, I can imagine. Only something a pharmacist would know, right? Something else I'd love to touch on is the recognition your work has received over the years. You've been honored with awards like the Nowitzki Prize and elected to the National Academy of Sciences. So when you look back on those moments, what did they mean to you personally, and how did they shape the way you thought about your work moving forward in the scientific field?
Um, Especially the National Academy of Sciences, that's a special honor,
Mm,
right? That's being elected by your peers.
yeah.
Um, so it's an incredible honor. It's probably the thing I'm most proud of. But it didn't really affect the way I do science. It
Mm.
didn't really affect that.
That's beautiful. These recognitions, I mean, hearing about them is really incredible, but what stands out to me even more is how many scientists you've mentored along the way. When you think about the students who've come to your lab, what inspires you the most about working with these young researchers that and watching them grow into independent leaders?
Well, um, being a professor is the best job in the universe. You realize the only person in the lab who gets old is me.
Oh.
Everybody else stays pretty much the same age. And so watch them go from real novices into very accomplished scientists is a great pleasure, actually. So I've had a couple students who have done extremely well, and a whole bunch of students who are still in academia at places all over the world, actually.
Wow. That's beautiful. I mean, is there a particular quality you've noticed in these researchers, something you wish more young scientists understand early on?
The most important quality is motivation.
Mm. Yeah, definitely. I feel like that's something that a lot of students lack these days, especially in this generation.
Yeah. I mean, you need a certain level of intelligence, of course, but
Of
motivation is
course.
really important because you have to deal with a lot of failure.
Mm. That's beautiful, yeah. You've mentored so many scientists who've gone on to shape the field in their own ways. From your perspective, what do you think is the most important shift that happens when a student transitions from just doing lab experiments to actually thinking like a scientist?
I think I think that happens in graduate school, actually.
Graduate school. Okay.
They start out usually they start out on a project that's sort of given them by the advisor.
Mm.
But oftentimes they branch out from that project and take off on their own.
Okay,
That's what the good ones do.
so what kind of separates undergraduate school from graduate school is kind of independent, leading your own path in a sense?
Yes.
Okay, I understand. All right. Diverting slightly, a lot of young people listen to my initiative, and this includes high school students who are just starting to get excited about science and research. So, what advice would you give them as they begin exploring research and figuring out whether this path is right for them?
Well, if you can, and I think you have, try to get some experience in a research lab.
Hmm,
So um, it gets easier when you're in college to do that.
definitely. Yeah.
Um, but that's where it starts for almost everybody, right?
Hmm.
You have to see if you really like it. And then you can see if you in college, if you get experience in a lab, then you'll get to see what graduate students and postdocs are like.
Oh, yeah. I this summer I visited Baylor College of Medicine and I did this very minimal research. Like I was just so fortunate to be a part of it at all. We did platelet cell research and I was kind of part of the process where you had to distinguish different parts of the cell. And I was analyzing rat cells and human cells. And it was just such an amazing experience. I mean, it got to know what it was like to actually work in a lab. I saw undergraduate students, postdocs, of course, the research professor was there. And it was just so cool seeing how, like, people can transition from regular biology high school classes to something that is life-changing and changing lives entirely out in the healthcare field. So I completely agree with what you said. I think live experience is definitely something that can help you get a real feel for what you plan on doing in the future. Okay, Professor Silhavy, we are down to our last 10 questions, our rapid fire questions. I created this rapid fire question set in a way where you can answer them as quickly as you can and as short as you can. It's just like a little fun, little like ping pong kind of thing. So the first question is, what is one piece of lab equipment you absolutely could not live without?
Centrifuge.
What is one small daily habit that keeps you grounded during busy research days?
I, I would say talking to the people in the lab.
Mm, like a little brain break.
Can I help with
Okay.
their problems? Yes.
What is your go-to brain reset activity when you need a break from the lab?
Reading.
Mm. What is the soundtrack of your workday? I mean, silence, music, or background noise?
Nothing.
Silence, got it. All right. What is the quickest way to spark your curiosity when you feel stuck?
The the quickest way is to show me a result that doesn't seem to make any sense.
Mm, interesting. If you could instantly master one technique that didn't exist when you started, what would it be?
um, I wish I knew more computer stuff.
If you were a scientist, what alternate career could you actually see yourself enjoying? And I know that's not a pharmacist for sure.
Um, you mean if I was not a scientist?
Not a scientist.
Does it mean I'm not a teacher either?
Mm, I mean, since you're already a professor, I guess not. So let's exclude that.
Oh boy, you're not leaving me very much choice.
Um,
Well, when I guess when I was a kid, I wanted to be an airline pilot.
Wow, interesting. Okay, what is one scientific dogma you were thrilled to see overturned?
The idea that we could improve the human race with genetics. Do you know what I
Wow,
mean?
interesting. Yeah, I see what you mean there.
I mean, like the Nazis tried to do. I mean, at that particular time, people really believed it, right?
Definitely. That's a good one. That's a good answer. What is the most surprising result a student ever brought to you?
Um, the This is hard to explain. There, there's a class. There was an example where the dominant suppressor we had isolated was recessive.
And our last question, if you could watch any historical scientist run a single experiment, who would you pick?
Francois Jacob.
Professor Silhavy, thank you so much for joining me today. It's been such a pleasure speaking with you. I mean, I've learned so much over the past 20-25 minutes. And I thank you so much for this opportunity, and I hope we can talk again soon.